Hi everybody and welcome to the walkthrough of the Biology Paper 1 Practice Paper 2. Now this is based on the advanced information from AQA, so it's only suitable for 2022. If we have a look at question 1, this is asking us about mitosis, cell division and stem cells. So the very first image we have here is a simplified version of the cell life cycle, the cell cycle. So in the first stage here, we're always going to have a replication of DNA. So that's always the first stage. Then we have the nucleus dividing. So the nucleus inside the cell divides. This is what we'd call mitosis. And then finally, we have the cell divides in two. So if we have a look at our diagram underneath, well, stage 1, we said it was replication, so copies of DNA are made. Stage 2, that's the nucleus dividing, and finally stage 3, that's the final bit where the cell itself divides into two. On to the second question. It says that the mass of DNA in a human cell at the start of the cell cycle is 6 picograms. What DNA will be in each of the new cells produced by the cell division? Now, we know that cells produced by mitosis are genetically identical to their parent cells, so they're going to have exactly the same amount of DNA. So if the parent cell had 6 picograms, then the daughter cell will also have 6 picograms. If this had been meiosis, then you would have had half the number of chromosomes, hence the mass of DNA would probably be half. So if it were meiosis, then we'd be looking at 3 picograms. On to a little bit about stem cells. So as it says in the question, stem cells are undifferentiated cells. This means they can become any other type of cell. If we have a look at these statements, we've got animal stem cells are found in meristems. Now, I know that meristems are the stem cells that are in the tip of plants, or the tips of plants. So I know that this one can't be correct. Animal stem cells divide by meiosis, and I also know this is not the case. Meiosis produces gametes. So we're not producing gametes in this instance, we're producing other types of cells, so it can't be this one. Meristem cells in the plant can differentiate throughout the life of the plant. Yes, I think this one is correct, but let's just check the last one. So meristem cells in plants can only differentiate into one type of cell. Well, no, that's not true. They can differentiate into any type of cell in the plant. So that is wrong, which means, yep, our first instinct was correct. It's this one. The final part of question 1 looks at therapeutic cloning, and I'll try and explain this a little bit with a diagram on here. So in therapeutic cloning, you will take an egg cell from a donor, so somebody has to donate their egg cells. You will remove the nucleus, so you end up with an egg cell with no nucleus in. You would then take the nucleus from one of the patient's cells and insert it into the donor egg cell, and that would give you an egg cell which has the DNA from the patient, and then you can kind of kickstart cell division using a little jolt of electricity, and then those cells will be genetically identical to the patient because you've taken DNA from the patient and put it in there, and you can then grow these into any type of cell you wanted. So the benefits of this, there's quite a lot. So you can use it to treat lots of diseases. You can produce replacement cells or organs. You are unlikely to have these cells, these organs, rejected because they have the same DNA as the parent's other cells, so they're not seen as invaders by the immune system. You can produce lots of cells, so many cells produced, and you can use these for research. Let's see if I can spell. And finally, you don't have a long waiting time for transplants. So you only need to come up with 2 of these advantages, but I've put all the ones that I could think of here. Disadvantages, obviously, if you were to let this modified egg cell divide and develop as normal, it could become a life. So we have the idea that a potential life is killed. You might also find there's a shortage of people who are willing to donate their eggs for these purposes. The eggs might be quite hard to get hold of. We don't know the risks. This is quite a new concept, a new idea, so we don't really know what might happen further down the line. You may transfer viral infections, and actually it's got quite a poor success rate, so you might have to go through several attempts of this to make it work, and again, every time you do this you're destroying a potential life, and you're using eggs that you don't have a very big supply of. Question 2 is about the heart and the effect of exercise on the heart. So we have a student measuring her breathing rate at rest, when walking and when jogging, and they've recorded their results in this table we have here. And the first question asks us to compare the breathing rates when doing the 3 activities. So this is a case of looking at them in relationship to each other, not just saying at rest we have 12 breaths per minute, walking we have 24, and jogging we have 60. We need to know that, we need to be able to read this off the graph, but then with that information we need to make a comparison. So if we look at this carefully, you can see between 10 and 20 there are 5 squares, so that suggests to me that every square is worth 2 breaths per minute, and this one is 1 square above 10, so that means we've got 12 breaths per minute. On the next one, we're 2 squares above 20, which suggests it is 24 breaths per minute, and this one is on the line for 60, so that is 60 breaths per minute. So we'll need to make a quantitative comparison of these 3 activities. So if we have a look at walking and at rest, so when walking, well, it goes from 12 to 24, so it's double the number of breaths per minute. So the breathing rates is twice or double that of at rest. So that's one comparison. Let's compare jogging and walking now. So 24 and 60. I think that's 2.5 times. So if you were to do 60 divided by 24, that's 2.5 times. So it's 2.5 times greater. So breathing rates when jogging is 2.5 times greater than walking. And the final comparison we need to make is between jogging and being at rest, sitting still. So again, if we were to do 60 divided by 12, that would give us 5. So the breathing rate is 5 times higher when you're jogging compared to at rest. So breathing rates is 5 times higher when jogging compared to at rest. So that's not too bad. Let's have a look at the next one. So explain why breathing rate changes when doing different activities. So this is all going to be to do with the energy requirement. So if you're more active, you need more energy. So breathing rate will increase, and that's to supply more oxygen. So we need to get more oxygen into our bloodstream, and we use that oxygen for aerobic respiration. So remember that aerobic means with oxygen, so we'll need oxygen to do this form of respiration. Aerobic respiration gives us a lot more energy and a lot fewer waste products than anaerobic respiration, which takes place when we don't have any oxygen. And that means we have more energy is transferred by the higher rates of respiration when we are more active. So key points to remember here: if we're doing something that's more active, we're going to need more oxygen to carry out more respiration. Because we've got higher energy demands, we need more energy. On to the next side. So we've got a simplified version of the circulatory system. We've got the left hand side of the heart here, which is connected to the body, and the right hand side of the heart here, which is connected to the lungs. So just remember that these diagrams are always opposite because you're looking at it front on. So in this image, the left ventricle and the left atrium are on the right hand side, so please don't be confused by that. Here we have the question which says the heart is a double pump. Describe what this means, and this diagram really helps us. So all we really have to do is explain what we're seeing. So the left ventricle, or the left side of the heart, so left ventricle, left side of the heart, that pumps blood to the body, and the right ventricle, or the right side of the heart, pumps it to the lungs. Pumps blood to the lungs. Next D, the wall of the left ventricle is much thicker than the wall of the right ventricle. Suggest one reason for this. Now, because the left ventricle has to pump to the body, it's got to pump further than the right ventricle, and it's also got to do it with a greater force and a greater pressure. So the the left ventricle has to pump blood further, more force, with more force, at greater or higher pressure than the right ventricle. And don't forget, this is a comparison again, so we're talking about the fact that the left ventricle is thicker, so we need to compare what it does to the right ventricle. And then finally, for question 2, people are encouraged to exercise after recovering from a heart attack. Suggest one reason for this. Well, any exercise is going to strengthen the heart muscle, so that's probably the most obvious answer. Another interesting point is that if you do gentle exercise, you're going to reduce weight, so it helps lose weight, and this will help circulation. So question 3 is about communicable diseases, and the very first part of it asks us, what does it actually mean for a disease to be communicable? And it's a disease that can be passed from one person to another. It's caused by a pathogen. So if we have a look at these, a disease that can be spread from one person to another, this is probably going to be the right answer. A disease that cannot be treated with antibiotics, I know that's not true. A disease that is not spread from animal to humans, I don't think it's going to be that. And one that is passed on through genes, I don't think it'll be that either. So we know this, and we've eliminated the other 3, so we're pretty certain that, yeah, this is the correct answer. A disease that can be spread from one person to another. First of all, we've got a woman becomes ill and she's got the following symptoms. She has pain when urinating and a thick yellow discharge from the vagina. So what disease does she have? Straight away to me this sounds like gonorrhea, but HIV, human immunodeficiency virus, again that doesn't match up with what we see here. Measles, you'll end up with kind of a rash. And salmonella is food poisoning, so these don't fit with this, so it's got to be gonorrhea. Next we have tuberculosis, which is a bacterial infection. That's probably very important to highlight. And we also, if you have tuberculosis, you get severe discomfort, so it's going to be painful. It's caused by a bacteria, so the medicine we can use to treat it or to kill that bacteria would be antibiotics. But remember that antibiotics will only work against bacteria. If the disease is caused by a virus, they're not going to have any effect. And what type of medicine should be used to treat the symptoms? Well, we've got this idea of severe discomfort being one of the symptoms, this pain. So to stop us suffering from pain, we can take pain killers, so aspirin, paracetamol, or even stronger ones. And last part of question 3 looks at how the human body defends itself against the tuberculosis bacterium. And there are kind of three overarching ideas here. We've got things that stop the actual bacteria getting into the body. So for example, skin, it's a barrier. We have blood clots. So if you cut yourself, your blood clots very quickly using the platelets. And stomach acids are methods that the body uses to prevent bacteria entering the body or getting into our system. So a few other things we could have mentioned there as well. We could have talked about the idea that your nose and your trachea has mucus to trap bacteria and stop it getting down. And you also have ciliated cells, or you have hair that helps move your mucus out of the body. But we'll leave it with these ones for the time being. So that's kind of the first line of defense. We've stopped the bacteria getting into the body. We've also got the idea of phagocytes. So these are non-specific white blood cells that engulf or ingest pathogens. This process would be called phagocytosis. They can also produce, these white blood cells can produce antitoxins. So what it is that makes you sick about bacteria is the fact that they produce poisons or toxins that damage your body. And if your white blood cells are able to produce things that counteract those toxins, you'll feel better sooner. And the last thing we have are lymphocytes. So specific white blood cells that produce antibodies. So we've got the idea of things that stop the bacteria getting into the body. We've got general defences which will ingest or engulf all pathogens, and then finally we've got lymphocytes that produce antibodies that will attack specific pathogens. And here we have question 4, which is about photosynthesis. So photosynthesis is the reaction that plants use to produce glucose that they can use for a variety of purposes, including respiration, storing as starch, making nitrates, making amino acids, and other things. So it takes place in green plants or algae. So you have carbon dioxide and water reacting in the presence of chlorophyll and sunlight to produce glucose and oxygen. The type of energy that we need for that, well, it's light energy. Usually, but not always, comes from sunlight. And what substance is it in the plants that absorbs this energy? So that's that pigment chlorophyll that I mentioned a few seconds ago. So it's the chlorophyll in plants that makes them green, and that's what absorbs the sun's energy, so it can be used in the photosynthetic reaction. In which part of the plant cell does that take place? It's named the chloroplasts. So if you see a cell with lots of chloroplasts, chances are it's going to do lots of photosynthesis. And we need a balanced symbol equation here for photosynthesis. I mentioned previously that it was carbon dioxide and water reacting together to give glucose and oxygen. So we'll start off with carbon dioxide, water. The symbol equation for glucose is C6H12O6, and also we produce some oxygen. So we've identified the correct symbols, but we've not yet balanced it because there are way more carbon atoms on this side compared to this one. And an easy thing to remember when you're doing photosynthesis is the devil's number. So think 6, 6, 6. So as long as you can remember these and 666, you should be fine. You could also take it a step further. You could talk about the fact that carbon dioxide is a gas, water is a liquid, glucose is dissolved in water in aqueous solution, and oxygen is a gas. Next one asks us, describe 2 ways that you could speed up photosynthesis, and this links to those limiting factors of photosynthesis that you've probably spoken about in lessons. So you could increase CO2 concentration, you could increase light intensity, and you could also increase temperature up to a point. If you go above around 40 degrees, the enzymes in the plant start to become denatured, so actually the rate of photosynthesis would decrease. And the last part of question 4 shows us the cross-section of a leaf, and it's identified 2 cells. This first cell, which is kind of on its ends and it's packed very tightly near the top of the leaf, these are the cells that carry out photosynthesis. So these are the palisade mesophyll cells. So palisade mesophyll cells. I'm going to put the mesophyll in brackets because you don't necessarily need to say mesophyll to get the mark. As long as you identified that it's the palisade cells, you should be fine. So 1, those are our palisade cells. And because they're right by the surface of the leaf, they're going to have lots of chloroplasts for photosynthesis. So lots of chloroplasts for photosynthesis. And the next cell is the guard cells. So these ones here, either side of the stomata, are called guard cells, and they can open and close the stomata. To allow CO2 in. Oops. or oxygen or water vapour out. Question 5 looks again at photosynthesis. This one is actually focused on the required practical that allows us to measure how those limiting factors can affect the rate of photosynthesis. So we're looking at light intensity this time, and we've put a lamp near some water with some pondweed in. We've got a meter rule. We can change the distance between the two. Moving the lamp further away will decrease light intensity, so in theory should reduce the rate of photosynthesis. Counting the number of bubbles in 2 minutes, so that's the way we measure photosynthesis. So make sure you're always specifying how long you're counting the bubbles for, and then you're changing the distances from the pondweed. Okay, that makes sense. The students could not make a firm conclusion because their method did not control enough variables. So if you're controlling variables, you're keeping them constant. And unless you keep your control variables constant, you can't have what's called a valid investigation. So you can't trust your results. So 2 variables that they did not control that would affect the rate of photosynthesis. And we'll go back to those idea of limiting factors. We're changing light intensity. That means we'll need to keep carbon dioxide concentration and temperature constant. And they've not done that. So we'll start off with temperature. The problem we have with using a lamp is that this will emit lots of heat energy. So by moving it closer or further away, we're also changing the temperature. If we wanted to, we could use an LED lamp that gives off less heat to control temperature. So we're not really asked about it in this question, but if we are, then if we wanted to control temperature, we could use an LED lamp. And for the other one, we've also not controlled carbon dioxide concentration, so CO2 concentration. And again, if we'd wanted to do this, we could have used a chemical like sodium hydrogen carbonate. If we'd have added that to the water, that would have controlled the amount. We could measure the amount of carbon dioxide we put in there. So that would have made it a better investigation. Underneath we've got a table of results. They've calculated the mean, and they've missed out this one. So I think this is what we're probably going to have a look at. So here it says calculate the mean rate of bubbles produced per minute when the lamp was 10 centimetres from the pondweed. And again, we have to read this carefully. So let's go back to our table. And we're looking at the one where the distance is 10 centimetres away. So we've got 22, 34, 31, 31. Now, when we're working out a mean average, we only use concordant data. So if you look at these 3 values, they're quite close to each other. However, this one is way below the other 3. So I believe that this one is going to be an anomaly. And we don't use this one when we calculate our averages. So we're going to take the average of these 3 values. So 34, 31, 31. So we had 34 plus 31 plus 31. And again, we're going to be quite extra about this, and we're going to write down every stage of our calculation so we maximise the number of our marks. So 34 plus 31 plus 31 equals, that's 96. And I then divide that by the number of readings I've used. So 1, 2, 3. So I've used 3 readings, not 4. I'll divide that by 3 and it gives me 32 bubbles per minute. Per 2 minutes. And we have to be careful because it's every 2 minutes, but the question is asking for bubbles per minute. So I'll need to divide this number by 2 again. equals 16 bubbles per minute. Again, be really, really careful. That almost caught me out there as well. So don't forget that in our table, it was number of bubbles in 2 minutes, but the question is asking us for how many bubbles we had in 1 minute. So easy to make that mistake. The mean number of bubbles released when the lamp was 30 centimeters away from the plant was greater than when the lamp was 50 centimeters away. How many times greater was that? So we're going to have to go back to our data, and we're looking at 30 and 50. So at 50 it was 6, and at 30 it was 14. So we're going from 6 to 14. And if I want to know how many times greater it was, I just do this number divided by that number. So the big number divided by the small number, which I think will be 2 point something, but we'll use our calculator. 2.3 reoccurring, and I'm not going to use the reoccurring. I'm going to round it to 1 decimal place. So again, we're not going to use that recurring. We're just going to do 2.3. The student wants to find out if different wavelengths of light affect the number of bubbles released. So the wavelength of light controls the colour of the light. So all we'd have to do here is use different coloured filters to change the colour or the wavelength of light. And to make sure we're keeping control variables constant, we'll keep the lamp the same distance from the pondweed each time, so that keeps intensity the same. So control light intensity. By keeping the lights the same distance from the pondweed. So question 6 looks at the digestive system and also at food tests. So the first thing it asks us is to name the enzyme which digests starch in the human digestive system. So the enzyme which breaks down starch into simple sugars, into glucose. And we know that is amylase, which is a type of carbohydrate. And we have an example of an experiment here which should model or represent how the human's digestive system works. So we've got a water bath at 37 degrees Celsius. This keeps everything at body temperature, so it's similar to the human body. You've got the water outside the whisking tubing, which represents the bloodstream. Inside the whisking tubing, which is inside, it's a model for inside the intestine, you have the food and you have the enzymes that are breaking them down, and then the partially permeable tubing itself represents the small intestine, so the lining of the small intestine. So again, this food inside will start to be broken down by the enzymes, and if it's able to, it will pass through the partially permeable membrane, it will pass through the small intestines, and it will be absorbed into the bloodstream, where the bloodstream takes it wherever it needs to go. Again, I'm not going to read all this out. I'll let you have a look at that yourselves as you're doing the question. B suggests which part of the body the partially permeable membrane or the tubing and the water in the test tube represent. So that whisking tubing, that partially permeable tubing over here, that represents the small intestine, as we mentioned previously. and the water in the test tube. So that's what all the nutrients will be absorbed into. So as we mentioned earlier, that's an analogy for the blood. The table underneath gives us the experiment results. So inside the tube right at the start, we've got starch, that's what we put in, and we've got no sugar. And we've got no sugar because the enzymes haven't started to work yet. Well, they haven't broken down any starch yet. In the test tube, there is neither starch nor sugars, so nothing's passed through there yet. Number 3, after 30 minutes inside the tube, we've still got some starch, but some of that starch has been broken down into sugar, into glucose, by the enzyme. So some starch broken down into sugar by the enzymes, but it's quite clear that not all of it has been broken down, so there's still some starch left. And at the end, the water in the test tube still got no starch in, but it does have sugar, and this is because sugar is a small molecule, so it's small enough to pass through the whisking tubing, through that membrane, but starch is too big. So your starch is too big. So that's kind of explaining what's happening here. So let's see what they're going to ask us. The reagents used to test for starch and sugar is the first thing they're after. So starch, we use iodine, and it's not asking us, but that will go from an orangey brown colour, and if it's a positive result, it will turn a blue black colour. Sugar, to test for sugar, we would use Benedict's. So just use Benedict's solution, and it's worth noting it has to be greater than 60 degrees Celsius for this to work. You will notice that it stays blue if it's a negative test, but if it's a positive test, you'll end up with either a green, yellow, orange or red colour being produced. If there's lots and lots of sugar, it goes red. If there's a little amount, it goes green, and if there's none, it stays blue. Other tests you will need to know about are for fats and lipids. You would use Sudan 3, which would give you a floating red immiscible layer on the top if it were positive, and Burette's will go from blue to purple if you had protein present. Why was there no sugar in test 1? So just like we mentioned earlier, so in test 1 we said the enzymes haven't started to break down the starch yet, so the enzymes hadn't started to break down starch. Not started to break down starch into sugar. For E, it says explain the results for test 3. So this was the most complicated one. So this is where we had both starch and sugar, and we said that some of the starch has already been broken down into sugar, but it hasn't done all of it yet, so we've still got some starch left. So so the enzymes have broken down some starch into sugar, but not all of it. And finally, question F, which asks us to explain the results for test 4. And let's have a look at test 4. It's the one where we're looking at the water in the test tube, so that's analogous of the blood, and there is no starch but there is sugar, and we said that's because the sugar molecules are able to pass through the tubing but the starch molecules are too large. So sugar molecules can pass through the tubing, but starch molecules are too large. And the last question, question 7, focuses on the microscopy experiment and cell structures, no pun intended. So over here we've got to start off by drawing one line from each cell to the type of cell where the structure is found. So nucleus, that's only found in animal, plants and fungi cells. So those are eukaryotic cells. So eukaryotic cells have nuclei, whereas prokaryotic cells, their defining feature is they don't have a nucleus. A permanent vacuole you will find in plant cells only. Plasmid DNA you will only find in prokaryotic cells, in bacteria cells. So we could summarise that with, so it's a simplification, but these have no nucleus, whereas these will have a nucleus. Onto here we've got a plant cell, and we've got 3 structures labelled. Let's see if we can figure out what they are. So this large one in the centre, this reservoir of cell sap, I think we can identify that as a permanent vacuole. This tiny structure here, B, that's too small to see with a light microscope. You'd have to use an electron microscope with a higher magnification to see this. I think this one is a ribosome. And then finally, I think we're pointing at the cell wall here because it's the outer layer. So let's label that cell wall. And I think the most confusing thing about this is actually the layout of the question. We need to figure out which of these rows is correct for structures A, B and C. So my first structure I'm pretty confident was a vacuole. So the only 2 options with vacuole for structure A are these 2. So it's got to be one of these 2. Next, I thought this was a ribosome. Go over here. Yep, ribosome. So we're looking pretty good for the last row. And finally, we'll check cell wall. Cell wall, good. Yeah. So I'm confident that this is the correct row. Over here we've got a student observed slides of onion cells using a microscope, and this is what they saw. So you can't really see what's going on here. So what could we do to slide A, so this slide, to make it more clear? So we need to adjust the focus. This is a blurred image, so it's not in focus. So to deal with that, I would turn the fine focusing adjustment or dial to bring the cells into focus. So not too bad for that one. Next, as you can see, the cells are way too small to be able to make out the structures that we'd like. So we need to kind of increase the magnification here. So whatever you do, do not say zoom in, because the examiners do not like that. What we have to say is that we rotate the objective lens to a higher power. Higher power is absolutely key. Objective lens. So you'll recall that when you look at a microscope, you've got 3 objective lenses, and they just represent the different power lenses. So you'd want to move to a higher power lens where the magnification is higher. Next, we've got we finally managed to get a clear image, and we're looking at working out the magnification. So we know the image size is 112 millimeters, it tells us here. And we know that the real length of the cell was 280 micrometers, so that's real. And when we're working it out, magnification is always going to be image over the real size of the object. But one thing to bear in mind is that these need to be in the same unit, whereas over here we're not in the same unit. I know that micrometers are 1,000 times smaller than millimeters, so if I'm converting between the two, if I'm going from millimeters to micrometers, what I'll do is times by 1,000, or if I'm going the other way, I'll divide by 1,000. And I've got a choice here, so I can either convert this into millimeters by dividing by 1,000, or I can convert this into micrometers by timesing by 1,000. So I'll choose to times 112 by 1,000. There's no right or wrong method here. And that gives me 112,000 micrometers. Now all I have to do is substitute into my equation here 112 over 280. Looks like it's going to be quite a large magnification. We'll get our calculator. Let's turn it on, that might help. 280 gives me 400. And I'll write it in the correct space to make sure the examiner sees it, and there we are done.